Welcome to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey of KISS Organics. This is the podcast where we discuss the cutting edge of organic growing from a science-based perspective and draw on top experts from around the industry to share their wisdom and knowledge. It's been a little while since the last podcast. I wanted to let you know that I've been busy with some other projects, but also tracking down some new and exciting guests. I have some really great podcasts lined up in the coming month, including one with Allison Justice of Outco to help dispel some of the myths that have been floating around the cannabis cultivation industry for decades. And I know we've already done some podcasts discussing mycorrhizal fungi, including one with Jeff Lowenfels that I suggest you check out. I wanted to do this interview, though, to get a different perspective on mycorrhizal fungi from someone who has been involved in the industry and selling and using it for over a decade. I've had my opinions based on the research I've seen regarding topics like using a single species versus diverse species of endomycorrhizal fungi, as well as the addition of trichoderma to these mixes. Our guest this week is Jim Eagan. Jim has been working for Plant Revolution for more than a decade. Having been the first company to bring mycorrhizal inoculants to commercial applications, he and the company have been spreading the good news of the benefits of healthy soil to gardeners, landscapers, and farmers all over the world. His degree in environmental studies, his work in the permaculture community, and his experience running a small farm form a background that has given him a deep understanding of the importance of soil organisms for plant and ecosystem health. He continues to learn and explore the fascinating worlds of food production, native ecosystems, and sustainability. All right, thanks, Jim. I really appreciate you coming on the show.
Yeah, thanks, Dad. Really excited to be here.
So let's just start off and dive right in talking about mycorrhizae and mycorrhizal fungus. Would you go ahead and, I know we've talked about it with previous guests, but can you just give us a brief introduction into what mycorrhizae is for people that may be new to the subject before we dive into the little tougher questions?
Yeah, no problem. Essentially, mycorrhizae means fungus root, and it's a very important grouping of fungus that exists in the natural environment. And it literally is a fungus that lives on the roots of plants. So it has a really hard time producing its own food, namely carbohydrates and sugars. So it actually has partnered with plants, and it did this as far back as we can tell in the fossil record. So plants, when they first came on land from oceans, had very primitive root systems. And it was this coming together of fungus and ocean plant. You can think of it as like a holdfast for a seaweed, right? So the mycorrhizal fungi allowed much more surface area, so it's much finer than root hairs. And it basically would cover the arid land and absorb water and nutrients. It would break down minerals, all the things we're going to be talking about. But it covered these root systems and eventually formed with about 95% of the world's plants. And this includes, the early non-vascular plants from hornworts and liverworts and mosses to, the giant redwoods and conifers and flowers and vegetables. So almost all of the world's plants form with this soil fungus. And it's a little bit different from saprophytic fungi, which sort of exist independently and break things down in the forest. This stuff relies on a host plant. So that's a very brief overview. Any of those topics we could have spent many minutes on, but that's just a very brief overview.
No, that was great. I do want to highlight a couple other things too for people. So for those of you that are, I know this is a cannabis podcast, but we get a lot of people that are growing other plants too. Now, there's many different types of mycorrhizal fungi, which we can talk about in depth. The main ones being, Endo mycorrhizal fungus, Ecto mycorrhizal fungus, and then there's another one called Ericoid as well, which I always associate with azaleas and rhododendrons. But it's important to realize that there are different types of mycorrhizal fungus. Can you talk a little bit about what those types are and which plants they associate with, as well as the, things like brassicas that don't have that relationship?
Yeah, absolutely. So, like you said, there's a couple broad groups. There's the endomycorrhizae and the ectomycorrhizae are probably the two biggest. They also have things like ericoid mycorrhizae, and even orchids have their own sort of partnering mycorrhizae, interestingly enough. And really the two main types that commercially and practically we concern ourselves with are these endo and ecto. And so endo mycorrhizae is what we're all familiar with. It's also called arbuscular mycorrhizae or VAM, vesicular arbuscular mycorrhizae. Those are two structures that it forms, vesicles and arbuscules. And that forms with roughly about 85% of plants. So that's the type that forms with cannabis, it forms with vegetables, it forms with fruit trees. And really that's the main type that we deal with in commercial uses. The other type is called ectomycorrhizal fungi, and this forms mainly with your conifers, your oaks, other hardwoods and things like that. And that forms with roughly about 10 to 12% of the world's plants. It's much more ubiquitous in the environment. So I was on a conference call earlier this morning, just kind of walking around the garden outside here, and I literally found a giant fruiting body of one of these ectomycorrhizal fungi. So they're everywhere. You're probably breathing in the spores right now. But those, so that forms another big portion of the mycorrhizal fungi out there. And then, like you said, the Iroquois form with, kind of your acid soils, your acid-loving plants. So, cranberries, huckleberries, rhododendrons, things like that. And then we also have, those, they're not as stable and easy to produce, and also that's a much smaller grouping of plants that they form with. So as far as I know, those inoculums aren't very available on the market. Same thing with orchid mycorrhizae. Now I'm sure orchid growers probably have access to that, or they culture their own, But generally those two types really aren't available on the market, either retail or commercial. And then, the plants that don't form, which actually there are a lot of weeds. So there are a lot of the sort of plants that thrive in disturbed soils, right? So they're the pioneer plants, as we call them in permaculture, and they come in and they don't need a lot, right? They can survive. They can break through concrete, and you've seen like a dandelion, things like that. So these are plants that have evolved and just really never had a need for this mycorrhizal symbiosis, which is a really interesting thing. But yeah, there are a small grouping of plants, and luckily for gardeners, a lot of them are weeds that don't really need a mycorrhizal relationship. And in fact, mycorrhizae actually helps deter these a little bit in one's garden. So, that's kind of a broad overview of all the different types.
Now, if we were growing something, though, like cauliflower or broccoli, which does not have a mycorrhizal relationship, aren't there studies that show it actually will potentially be detrimental to the plant if it does form that relationship? So we want to be careful in how we're applying it into which plants?
To my knowledge, they don't form that relationship. So it's not really like a fungal forcing onto the plants. It's the plant really chooses which mycorrhizae to let in. And in fact, plants can actually turn off and change the mycorrhizae that they're forming with throughout a season, the same plant, to get its needs met. So, if it needs, more micronutrients, there's species that are more adept in getting, maybe zinc or something like that out of the soil. And so the plant will actually shift the mycorrhizae that it's forming with. So, the only thing that I can think of is that in a heavily mycorrhizal dominated soil, if you have other plants growing with mycorrhizal fungi, maybe it's out competing the brassicas, but I've never heard of it being detrimental to brassicas. That would be a new one to me. But
okay.
Yeah, I'd love to learn more about that.
Yeah, no, I thought I had read a study where it showed that it is possible to form that relationship, but it's actually detrimental to the plant. But it's not a natural association. It was a long time ago, and I could be wrong. I haven't brushed up on it in quite a while, honestly. So
right. No, look into that. That's interesting.
If you do find out anything, let me know. One thing you did bring up, though, that I wanted to go back to is you mentioned you saw a fruiting body or a mushroom of an ectomycorrhizal fungus. And that's something that I see with a lot of growers. There's a couple things out there where you see photos of roots and people are, let's say, let's use cannabis in this case, and they may see a bunch of mycelium and say, oh, that's my mycorrhizae, or they'll see mushrooms popping up in their, indoor cannabis garden, and they'll say that that's because they have a mycorrhizal relationship. Now, as I understand it, feel free to correct me here, the endomycorrhizal fungus, you can't see without special microscope tools to dye the roots to see if they're actually infected. And then those particular glomus species don't necessarily form fruiting bodies or mushrooms. Is that accurate?
100%. Yeah. Yeah. So what they're seeing is probably some saprophytic fungi that has found its way, and it's breaking down organic matter. So the most common one is the flower pot. It's a little yellow mushroom, and what's it called? Flower pot. I forget the exact species, Lucidium or something like that. And it's just pot culture conditions. So growing plants in soil and pots is just the ultimate environment for this stuff. So it hitchhikes in on a lot of, maybe it was probably in the soil to begin with, or just finds its way in there. And that's coming up in my house plants right now, but we get guys that will send us pictures of that. And no, that has nothing to do with the endomycorrhizal fungi. You're correct in that they don't form fruiting bodies, which is why, and we'll get into this, they're so little studied. They really are. If you look at the studies, it's like 2 or 3 species at tops. And, the estimated numbers at this point is over 300. And that's all the ones we don't know about. So we've only studied a small portion of them because they don't form fruiting bodies. So essentially, they have to, isolate these spores. They have to pull them out of soils. They have to, they can pull some of the mycelium, which, like you said, is smaller than is visible to the naked eye. You can see ectomycorrhizae, as it forms these things called rhizomorphs, which is like a lot of strains of mycelium kind of coagulated together. So you can actually see that, which is that famous picture. If you've ever Googled mycorrhizae, it's different species of pines, but on their root systems, it's this big sort of hairnet of mycelium radiating out in all directions. So you can see that because that's an ectomycorrhizae. But you're correct in that these mushrooms are not connected to the mycelium, and growers, you won't really be able to see the mycelium with the naked eye. Now, you will probably see a strong, healthy root system. We can talk about that later. But just so there's some things that I can't talk about. We can't make any claims whatsoever about our products because we're not registered as a pesticide. But if you Google some of these species, you will see that, there are benefits from the fungus that when you pull a root system out and it's white, there's not, I should be very careful on how I say this, but there's not certain things. The color is the color that you want. The roots are vibrant. That indicates, oftentimes that it has a strong mycorrhizal relationship and everything's functioning as it should. All the micronutrients are making it up, and all of the, the different chemicals that the plant forms with that to protect itself are all functioning properly. But so you can't actually see the mycelium if you were to pull out a root system.
So, I think it's safe to say that plants that are healthier are less susceptible to diseases and other pathogens. So a plant that has a mycorrhizal association may, in theory, be a little bit healthier than a plant that doesn't, because most plants, like you mentioned, 95% of them want to have this relationship. And can you talk about some of the benefits and how they may differ between endo and ecto mycorrhizae? Because with the ecto, you're on the outside of the roots, like you mentioned, it's reaching out into the soil. The endo is infecting into the root itself. What are the differences in the benefits of these two types of mycorrhizal fungus?
Yeah, so generally with both types, you see increased surface area. So, like I said, the first coming together of these organisms was because plants' root systems weren't. The purpose wasn't mineral and nutrient absorption. It wasn't to break down minerals and nutrients. It wasn't to absorb water. They came from environments that those things were in, and sort of a kind of an aqueous solution in the ocean. So one thing you get is you get much more surface area. So the mycelium essentially radiates out. What you have right around the root is called the phosphorus depletion zone. So plants, when they're first given nutrients, they'll eat up all the phosphorus and nutrients right around the root zone. And so what these mycelium do is they sort of poke out through that. So they radiate through that, and they get much more surface area, able to take up those minerals and nutrients. You also see they release exudates in the soil. They release enzymes into the soil, which make nutrients that get locked up, but once again, bioavailable. So they make that available to the plant, the phosphorus and the nitrogen and the micronutrients. So yeah, there's a couple other benefits. So they can actually fix nitrogen, atmospheric nitrogen, just like the bacteria. There's some bacteria out there. We even, in our blends, we have bacteria that do this as well. It's not the rhizobium bacteria like in a legume. It's actually free living bacteria, but also the mycorrhizal fungi can fix atmospheric nitrogen, which people don't know. Not many people are aware of. That's one piece of it. So stress on the plant, transplant shock. When a plant's transplanted, it's sending out chemicals into the soil to actually germinate any mycorrhizal spores around it. When a seed germinates in nature, it's immediately sending out these signals because it's grown and or evolved, if you'd say, to have this symbiosis. So it's used to having these ambient spores around in its soil environment. So once this germinates, through all these mechanisms that I've been talking about and ones that we haven't mentioned yet, it allows the stress tolerance of the plant to go up. So too little water, too much water, variances in pH, variances in temperature, the plant's actually going to be able to withstand that and actually thrive through that a lot better than if you didn't have the mycorrhizal association. And that's really one of the benefits of mycorrhizal fungi, and actually one of the reasons that, and we'll get into diversity later, but one thing that these inoculums do is they allow you to bring your baseline up. So your worst performing plant does a lot better. Now your best performing plant, if everything lines up for that plant and your pH is right on, all your nutrients are right on, everything's perfect, and that plant does great. You're not going to get a 10% increase in yield, no matter what anyone tells you. But not everything's perfect. And really what these inoculums do, and what mycorrhiza's specialty is it brings that sort of almost like the law of minimums, where it brings your poorest performing plants up to a higher level. So overall, you're getting a more stable yield. You're getting a more consistent yield. And that's actually one of the reasons, just briefly, why we included diversity in our blends, is because specific mycorrhizal species perform different functions. So, like, I'll give you an example. Glomus clarum, one of the glomus species, it helps with dealing with excess salts. It improves mediation of heavy metal toxicity. Glomus deserticola comes from arid regions. It's better at drought stress. So let's see, Glomus massae, it reduces uptake of arsenic. It's one interesting example, but they all have these different sort of specialties. And so when the plant encounters something that it's detrimental to its growth, it literally will change directions and start to utilize whatever species that can perform those functions that it needs. Now, in terms of your question about endo versus ecto, to be honest, I need to research that. I've never actually been asked that. And I'd be really curious to hear the differences and the different benefits of the different sort of morphologies, how they work in the root zone, how that translates into the ecosystem. And one thing you touched on is, ecto grow kind of between the root cells, and endo grow into the root cell, and they form these little structures, which just kind of tell people this is why it's called arbuscular mycorrhizae, is that they go into the root cell, form these little structures that look like root systems, and that's actually where the water and mineral nutrients are traded to the plant through there's a small little membrane around the arbuscule, and in exchange for that, it gets sugars and carbohydrates that, like I said earlier, it can't produce itself through that structure inside the root cell. So I think of it as endo in. But in terms of the difference between the endo and the ecto in the benefits realm, I'd actually be curious myself.
Yeah, I think the important thing for people to keep in mind is that you want to make sure you have the right species for what you're planting. And if you're not planting conifers, do those ecto species have benefit? I know you and I have talked previously to this, and I've been a big proponent of single species inoculation, because all of the research that we see out there is primarily with Glomus intraradices, or Rhizophagus intraradices now, as they're calling it, and the Glomus mossae, or however you pronounce it. I think that was the researcher that discovered it. But those are the two most widely used and most widely available. And I'm just curious what research you've seen that would support using more diverse species of mycorrhiza in a mycorrhizal product.
Yeah, so the big one is endo versus ecto, right? Yeah, clearly if you're not growing ecto plants, there's not a need for the ecto. We have a number of products. We have a single species endo. We have a 4-species endo. We have a diverse multiple species endo and ecto. So we support all kind of directions that the market is going. But clearly you don't need ecto if you're not growing an ecto plant. The reason that we've included it in some of our blends over the years is it's, like I said, it's ubiquitous. It's literally, almost pennies per pound for us to include it. For example, in a gram of our popular product, there's probably about maybe like 250 spores of endo. Follow me now with this, right? This is 1 gram of material. You got about 200 to 300 spores of the endo. You have a couple million spores of the ecto. So they're much smaller than the endo, and they're super, super cheap to produce. They're literally, coming up. We harvest those wild. So we harvest those fruiting bodies wild. And what that did for us was allow people, allow our blends to be used in a diverse array of environments, the specific ones where we decided to include both. We made it really easy for the end consumer. And so, that was probably 10 years ago. We came out with those products, and sort of as the market developed and people were saying, we just want the endo and we just want the bacteria, we don't want everything included. We started to broaden our product offering. So we have products that are just endo and bacteria. And then, like I said, we have the just endo. So there's absolutely no detrimental effect to having the ecto in there. But like you said, if you're not forming with a conifer, it's not absolutely necessary. But it's one of my favorite products that we still do is the Great White in terms of bacteria and endomycorrhizae. Even if you weren't even, considering the ecto for the value that you get, the concentration of endo and the bacteria in there, is really unrivaled in the market. You'd really have to buy two separate products at, almost double the price to get those concentrations. But yeah, I mean, I get it. I totally get it. If you're not growing a conifer, there's no need for it. But yeah, they still prove to be super effective products if they do have the blend in there. But yeah, we support, one species, 4 species, 8, like the great white has 8 species of Endo. Now, specifically talking about Endo diversity, what our research is showing and what the research is kind of beginning to reveal, because it's in the past, it's basically this stuff is very little studied. Like you said, we have the Glomus irregularis, Glomus intraradices. You have Glomus massae, Glomus aggregatum. So you have a few species that have been studied, and really they've only been studied for a very limited kind of set of parameters, or they're not testing for. It's tough to test for all the conditions that are out there, all the reasons people might be using these inoculums in the actual field. And so, sort of the thinking, this has been passed on. I'm not the researcher. This has been passed on from sort of our research department and our scientists, and they're continuing actually to isolate different species and begin to test those for their viability in the market. And what was passed on to me is that having diversity, first of all, my ethos is diversity, right? So permaculture and ecology sort of dictate that as a principle, diversity is more resilient. So, and how that works out into the mycorrhizal sphere is just like I said, all of these mycorrhizal species have different specialties. So Glomus intraradices is known as a broad former. It forms early, and it's great at uptaking phosphorus. But that's not the only sort of problem that growers are facing. There's all sorts of conditions. You have pH problems, you have temperature problems, you have disease problems, you have sort of micronutrient deficiencies, you have nutrient sort of lockout, you have all of these things that the plant needs to, either the plant figures out a way to overcome these problems, or it shows up, as a reduction in yield or some sort of, we've all seen deficiencies in our leaves and those sorts of things. And it kind of talks about further, the plant's natural defenses, they actually rely on the ability to get micronutrients and to get these, just water and all of these other things to actually form these compounds and form these different chemicals and exudates and things like that, which can actually keep the plant growing to, maximizing its genetic potential. So with the mycorrhizal fungi, like I said, what we've seen is the plant is actually able to cut off flow of nutrients to specific species, and throughout the season, switch that over to other species that are actually taking care of and have the ability to solve the problem that the plant is facing. And that can happen throughout the course of a season, or that can just happen straight out of the gate, especially with this kind of moving more into broad acre applications. There's so many different conditions and environments that growers are facing, right? So different pHs, different soil makeups, right? Different soil chemistries, different climates, all those sorts of things. One mycorrhizal species is somewhat limited in its ability to overcome those various problems that the plant is facing, whereas a diversity of species does have the tool belt, sort of, or the tool chest, if you will, to solve those problems and create those compounds and be able to adapt to the situation. So that's really, like I said, we offer single species, but we promote a diverse species within those groupings, right? Endo versus ecto. So that's what we're seeing, and that's kind of how we've built our inoculums. But any inoculation is great. If you're using single species, multiple species, your plant's really going to see a benefit.
So you mentioned that a plant can switch its mycorrhizal association or infection mid life cycle. How does that work then? Let's take a cannabis cycle, for example, where you have a plant that may only have 55, 60 days of flowering time. Are you saying that the plant could start off with, say, a relationship with Glomus interradices or Rhizophagus interradices, and then switch halfway through to something else, say if conditions change, like to the Glomus deserticola, for example? Or can it have multiple infections within the same plant of different species at the same time on, say, different root hairs or root tips?
That's the information that I've been passed on, is that both of those scenarios can happen with mycorrhizal fungi. So, to my knowledge, that is possible with both of them. And like I said, all of this stuff needs to be studied further, and especially as cannabis is, we can actually study it now with labs and universities. But just the mycorrhizal discipline in general has a long way to go. So, like I said, inoculum, single species inoculum is fantastic for plants. As a principle, I think diversity is a good way to go. But we have a lot to learn in terms of how that functions in a root system.
Sure, I would totally agree with that. And there's a lot of challenges around organics and research. We were talking about this, I think, last podcast, but I know definitely off air, I was talking with a friend about this, how a lot of the research being done, it's very hard to do it with organics because there's so many variables and there's so much we don't know about the microbial interactions in the rhizosphere that it's very hard to isolate and make any definitive claims or evidence of claims moving forward based on what we're seeing, because there are so many things going on. There's relationships between different microbes. There's relationships between the plant and the microbes, and it's really hard to isolate down to the effect of one particular variable. But that kind of brought me to another question here. So one of the challenges as a consumer is there's so many different mycorrhizal products on the market. How do we evaluate them? And we talked just now, you explained your opinion on diversity versus single species, which made a lot of sense. But how do we evaluate these? Because there's a few different things I want to look at here. So we already talked about endo versus ecto. So if we're applying it to fruit trees, if we're applying it to our garden, we want to make sure we have endo mycorrhizal fungus in our mix. And that ecto is probably, it's not going to hurt anything, but it's not necessarily what we're looking for that inoculation. So we want to make sure we're comparing endo species. And then we need to look at, do we have a single species versus a diversity of species? And then finally, the concentrations, and this is something we should probably go over too, talking about how labeling works with propagules versus spores and what's most viable for consumers when they're applying these things. Because you see guys and they'll look at a label and they say, oh my gosh, this has 2 million propagules per gram. But when you break it out, it's actually maybe 250 propagules per gram of the actual species that could potentially infect or benefit the plant that they're growing. So can you just kind of maybe go over some of those things in a little more detail and explain your thoughts on that?
Yeah, fantastic question. So how I think of it is, this is going to sound funny, but we're exiting the age of the Wild West microbial inoculant. And what I mean by that is, we've had companies and products just claiming the most outrageous stuff. I won't name names, but those products are somewhat gone from the market. The super outrageous ones are gone, but the reputation of the company, the age of the company is a big deal because the stuff is microscopic. So you're essentially buying a car that's under under a blanket or something like that. You're not allowed to look at the car. You're not allowed to inspect the car to see the integrity of the engine or the wheels or anything like that. So this stuff is microscopic. So, the company's reputation in the industry makes a big difference. And the first thing, so, when you go and take a look at these inoculums, the first thing you want to do is flip it over and start with what they're claiming. We'll get into that, whether or not that's reliable in a minute, but start with what they're claiming. So if you're looking at, apples for apples, you're looking for Endo, look at the diversity, and then look at the propagules. It'll either say propagules or grams, or propagules or spores per gram. And that's a good indicator of how many, units of material are in that will actually germinate and create this mycorrhizal network. And so what propagules and spores is that, so spores are it's like the seed of the apple, right? So you can grow this fungus with a spore because it has the genetic material, just like a seed. And the term propagule, as we were talking about earlier, it would be easier with fruiting bodies and mushrooms who produce much more even spore material. They're actually spores, but with the endomycorrhizae, because they're underground. So think of that, how that functions in an ecosystem, right? You have plants that die, that their root system is where these spores were formed and where these spores are going to pass on that genetic material. So oftentimes the spores don't have the advantage of, hanging from a cap as a gill would in a mushroom, and it can be blown around by the wind, or it can be knocked by an animal, or oftentimes squirrels are really important, especially for truffles with distribution of these organisms. You don't have these mechanical means of moving these spores around the forest, so oftentimes the spores end up in the roots themselves, and the plants just die, and that slowly decomposes. But that root is actually still viable because it's either filled with spores or it's just a fragment that will actually germinate into, when the next plant root finds its way down there, sends out those chemical messengers we were talking about earlier, then that can germinate. So when you're talking about production of endomycorrhizal fungi, you're going to end up with both root fragments and spores, and they're both viable. And, there is a process that you can just kind of extrapolate the spores, but it's very expensive at this point. So when people claim, oh, we've got this and that, billion spores per gram, it's not accurate. If they're going to have any price that's somewhat competitive in the marketplace at this point, they probably don't understand their product as they should. But so usually it's going to say propagules. That's a more accurate description of the material that's on the market right now. And like I said, you want to look at the concentration. So 200 to 300 propagules per gram is very, very high. Is very, very high at this point. That's a real concentrate. Around 100 is a good, is not bad. That's kind of what our lawn and garden mixes are, just because, the average person buying a rose or a tomato plant is not going to go for the higher prices, because when you add those concentrations, the price goes up. So, we could sell you a 4 ounces of material for $7,000 or something like that, because you can get these things really concentrate. But so, what you're looking for is a good 200 to 300 propagule per gram material. Our liquid, however, with milliliters, it's a little different. It's a little different volume. So we're close to, I think, like 60 to 80 propagules per milliliter. And how that works out is like, it's the same rate or the same cost per plant to use, even though the numbers are different. The way that the volume works out, it's the same. If you were going to treat a 4-inch plant, it would cost the same with our liquid as it would with the great white, or our powdered products. And then you can go with the straight mycorrhizal inoculum. we have products like that. A lot of our products, like I said, we pride ourselves on diversity, also carry specific bacteria that have been well documented and studied to provide things for plants. So, for example, nitrogen fixation or phosphorus solubilization, enzyme production. So all these things that work hand in hand with the mycorrhizal fungi and all the other soil organisms for that matter. But these secondary metabolites, as they're called from the bacteria, are super functional and helpful for plants and mycorrhizal fungi. So if the inoculums have some beneficial soil bacteria in there, we found that the synergistic effect of mycorrhizal fungi combined with these bacterial concentrates shows better yield or better results versus just using one of those individually.
Okay, so essentially as a consumer, what you want to look at is the concentrations, which will most likely be expressed in propagoules per gram, of the particular species which we're looking for, or type of mycorrhiza, which would be Endo, and then beyond that, diversity, like you said. And then lastly, one of the things that you touched on there that I want to examine a little closer is that there's no one really doing research or testing of people's claims, as I understand it. I know with soil, like I had to have, when I registered my product, I had to make a guaranteed analysis claim if it's going to be registered as a fertilizer, and then whatever that claim is, I have to show a lab test supporting that. But then at that point, it's the state would be randomly testing the product, maybe pulling it off the shelf to see if it actually met those standards down the road in terms of in the actual bags themselves. So I assume it's the same on the microbial side. Like if you had one test, for example, that said you had 1,000 propagoules per gram, you could put that on the label, and then in theory, until the state or whoever registered body tested it, no one would ever know what was actually in that product. Am I saying this correctly? I feel like I know there's been some tests done by the Oregon Department of Agriculture that have shown mycorrhizal levels and other microbial levels from 2016 as being very different than the claims that were on the label. Can you touch on that at all, or is that too tricky of a subject?
No, absolutely. And that's we're actually really happy that that's happening because, like I said, that gives companies like us who have been, honest and, actually putting in what we claim, a huge advantage. So, yeah, Oregon is beginning to test microbial products. And, they're not perfect. I think we actually, my family's company sort of helped them develop some of the tests for the mycorrhizal fungi this is, 10, 15 years ago. So, mycorrhizal fungi are much different than bacteria, and they were sort of treating them like bacteria. And so, yeah, the testing is beginning to happen. And that's kind of why I said we're coming out of the age of the wild, wild west, because there were some products out there that just made outrageous claims. And this testing is going to consolidate and kind of weed out some of those companies and products who who aren't being truthful. So, yeah, that's absolutely accurate. Like you said, people can put whatever they want on. We're responsible for a guaranteed analysis in terms of NPK and, derived from statements. But with the microbials, it's kind of been this gray area. And Oregon's kind of leading the pack. But a lot of other states will probably adopt some of the testing methodologies and the regulatory piece of that. So it's a good thing for consumers. And what we do, this is kind of how it works on the microbe side, is so we'll actually, we'll formulate things to concentrations that we think are adequate for, the type of grower that we're making the product for. And then we'll actually get it tested by an independent lab, and they will verify what we've been doing and aiming for, and then we'll reduce it by about 20 to 25% when we claim it on the back of the bag, to give a window so that when they do, because the regulators will go through and, as you know, just pull a product off the shelf at any licensed retailer and test it. And so we actually allow we claim less than we know is in the bag for the odd batch that doesn't have didn't get mixed perfectly or whatever. So we actually claim a lot less than what's in there that we aim for. And the fertilizer is the same. People do the same thing with fertilizer. But yeah, that's kind of the state of the testing, and it's a really good thing for consumers, because these microbials, like I said, are invisible. So people could claim anything and it would never be tested.
So the best thing for people to do is to take a look at a label, try and understand it to the best of their ability, do their own research, and then really just look at plant response. So I think, use Great White, use some of these other brands, and see what kind of results you get. And that's ultimately what we're looking for anyway, I think, is plant response. So that's
absolutely, yeah. Be in touch with your garden. And that's really why we've been successful as a company, because people do these side by sides. I can't tell you how many times I go to a new nursery, or we hand out a sample at a trade show, and we'll get pictures back like, wow, look at the difference in the root system, because this is an ancient relationship. Plants depend on this. It's like going through and bleaching the inside of a human being. You'd only last about a week or two. So it's almost how we've been treating plants, is we just destroy the biosphere and expect these plants to perform, at a high level, but then we end up, okay, well, now we need like 90% extra fertilizers, because most of it's getting locked up and the plant roots can't release the exudates to get it unlocked, or we hit them with fungicides or pesticides because now their immune systems are compromised. Like, it's a no-brainer. These microbes work. Like, mycorrhizal fungi works. It's a relationship that's, like I said, as old as land plants. So use the stuff and see it for yourself. And until you actually try something and see it, it's all hearsay anyway. So don't take what I'm saying as the truth. Try it out for yourself.
Great. Now I want to talk about some of the other bacteria and other ingredients in Great White. But before we do, can we talk a little bit about best practice? So what is best practice around application of these microbial products? Because one thing I want to touch on, because we were just talking about labeling, now that I'm thinking of it, is I see you'll get a soil bag or a bag of fertilizer and they'll say with mycorrhiza on there. And then you go and look on the back and it's like 0.008 propagules per gram or something, like just a really, really small number. And I typically tell people that that's probably not enough for them to get mycorrhizal infection. They're better off applying it directly to the roots of their plant rather than applying it, homogeneously throughout the soil or trying to get it with fertilizer. And as I understand it, because it is a sporopropagule, it also doesn't move through the soil very effectively. It needs a physical mechanism of watering or something like that. So we want to apply it directly to the roots. But can you give what your pointers would be for people who want to use, a mycorrhizal product with their plants?
Absolutely. So, yeah, direct contact is essential. The plant root releases a chemical, I'm forgetting the name, that triggers the spore to send out a hyphal tip, which then plugs into the plant itself. So that physical contact is crucial. When you have microbes that are dispersed, a foot away, 3 feet away from the plant, if you're doing a short season annual crop, most likely it's not going to germinate until late in the season, and by that time it really is not benefiting the plant. So you got to think of all those spores as investments. You spent money to get those into your soil. The best way to use them is to get it right on the plant root. So we have powders that you can sprinkle right on the roots themselves. You can also dissolve those powders in water and water it in. You can send it through your fertigation system, put it in your reservoir. have it drip down in there. So any way for it to actually make contact with the root system is beneficial. We also have granular products that were designed not to be dissolved in water, just to be sprinkled into the hole at transplant time. So that's really the most cost-effective way to inoculate your plants. And to talk about bagged soils and fertilizers, yeah, I think it's a good thing that people are paying attention to, and soil companies, fertilizer companies are beginning to honor the soil microbes and include them. The problem with soil is that, like you said, the concentrations are really, really low. So to see a good plant response, for example, our bare minimum for a 4-inch plant is about 10 spores if you're talking about Endo mycorrhizae, 10 spores per plant. So oftentimes with these, bagged soils, you'd have to use a whole lot, or there's just not enough material in there to actually make a real difference with the plant. Besides that, these bags are often stored in direct sun. So if this bag sits in the sun for a day, most likely everything in there is dead. If something does happen to get in there that you don't want, so once again, or I haven't even talked about this, but the concept of having a sterile soil, the problem with that is that is a ripe environment for an explosion of some sort of disease or something that you don't want in there. So one of the real ways that these inoculums work is that they create this balance. They create sort of this net that when something unwanted comes into the system, it's immediately outcompeted. That's a huge one with the mycorrhizal fungi, is that there's not a lot of ambient nutrients or spaces for disease to exist. So that's a big one with the soils, is that they often get sterilized in the sun, besides having low concentrations. And with the fertilizer, it's generally the same thing, really, really low concentrations. It's too expensive for them to really hit it with a lot of the sort of microbes. So what we recommend is buying straight fertilizer, buying your straight soil, and then adding the biology when it makes it to your site. And another reason is that we as a company pay a lot of attention to and spend a lot of time and effort in maintaining an environment and shipping our products and educating the stores and the retailers on how to store these products. It's printed right on our bags. The bags are all designed, as well as the jars, for sort of maximum stability of these organisms. So also in the supply chain, the delivery, these organisms are taken care of so that once they make it to you, they're in a prime condition to come out of dormancy and colonize your soil and your root system. So yeah, those are key points.
So for a fast crop like cannabis, for example, that may, maybe 90 days, for example, or even less in a lot of cases, we want to get the mycorrhizal infection to occur as quickly as possible, which means directly onto the seeds or rooted clones when we transplant. How important is it to re-inoculate when we transplant at later stages? Because I've always been a big proponent of doing a really heavy inoculation right at the time of planted, say your rooted clone or your seedling, and then not reapplying when I transplant at later stages, just because it gets so expensive relative to potential benefit. Have you seen benefit with later applications of mycorrhizal fungus at other stages of transplant?
With a pure mycorrhizal inoculum, I would say that what you're doing is fantastic. Maybe one more application. With our products, they also contain huge diversity and really high concentrations of the beneficial bacteria. Not talking about our products, but talking about BT, Bacillus thuringiensis, which our products happen to contain. That's a biopesticide in the organic world. So it's almost, so I think of these inoculums that contain the bacteria as well as the mycorrhizal fungi. It's basically instant compost teas. So I'm always revolving. Sometimes I'll just use our straight powders in water, or sometimes I'll brew compost tea with worm castings or compost. And I'm constantly adding T's in biology to my soil. But in terms of a straight mycorrhizal inoculum, I think that one heavy inoculation will give you a really good colonization of your root system. Doing it more, it depends on the specific growing conditions. But people will have to try it for themselves. As a general rule, with a straight mycorrhizal inoculum, I don't think you're really going to see a huge return on investment for a second and a third application. But like I said, our products are a little more complex. We have seen people using it every feeding, every watering as a preventative. So, whether that's broad acre or you're growing in a very, controlled environment, you're growing indoor, every condition's different. You'll kind of have to assess it for yourself and see if that prevention is necessary and worth it for you, or, if you're getting along just fine and you don't need it. But it's essentially like taking a probiotic, so people take those ongoing. And with the mycorrhizae, really what you're looking for is just to get around, 75 to 85% colonization of the root system. That's what you're aiming for. So it depends on the success of the first inoculation, which, is different every time. But if you're doing it, like you said, a really heavy inoculation right on the roots, you're probably getting a really heavy colonization.
Now, how long does that time to infection occur, or colonization as you're calling it? My understanding is it takes at least 10 to 14 days under good conditions, or even longer. Is that accurate, or what are you finding for infection rates?
Yep, that's accurate. We see the bacteria in our products go to work a little quicker. So like the great white has glycine, which is a simple sugar, B1, vitamin C. And we see people like, oh my gosh, my roots are going nuts. Like they're super white, or it helps remediate something that's gone awry. Generally, that's the bacteria and like the trichoderma, which I think we'll probably talk about next. But those are heavy enzyme producers. So they're breaking down organic matter. You've got dead roots on there that are turning brown. You've got sloughed off roots. You've got excess, organic matter in the root system. Those bacteria are just really, really heavy, like I said, enzyme producers, micronutrient solubilizers. So they're really going to work and just cleaning up that whole root system. So yeah, generally mycorrhizal colonization takes just under 2 weeks, and the bacteria sort of peak, and then the root system returns to a kind of a steady state based on the exudates and the sugars released by the root system itself. So bacteria peak, and then mycorrhizal colonization takes about 10 to 14 days, like you said.
Okay, I do want to talk about the bacteria, but I had a couple more questions before we do. So
yeah,
one of the questions is the persistence of these mycorrhizal propagules or spores in the soil. Like, for example, if someone was running a living soil indoors, or even let's say soil in their greenhouse or outdoors, they pull the plant, or even just cut the plant at the stalk and let those roots break down right there in the soil. What is the persistence of that those mycorrhizal spores? And do we need to re-inoculate every cycle in, say, a living soil bed or in soil that we're reusing?
Yeah, so, what I generally tell people is that I haven't seen a ton of studies on carryover of roots into, season after season. I guess it's fair to assume if it's the same species, you will have some carryover. I don't know. So like a lot of these spores, or a lot of these endomycorrhizal root systems don't, some of them take, it depends on the species. I mean, it depends on the growing condition. So some of them can take 3 years to produce spores. Some of them can take, 6 months, 7 months, like one growing season. But I'm not an expert in that. It's plausible that the roots of, will stay alive long enough, or the mycelium will stay alive long enough to colonize the next crop. But I don't think there's a whole lot of research done on that. So I wouldn't really be confident or comfortable making a recommendation. With the cost of inoculation and the business that we're in, it's a no-brainer for me. It's like if you're going to buy some fish and some kelp, and rock dust, which I advocate you do, why not buy $20 of material to treat your whole crop with mycorrhizal fungi? I mean, you don't have to use this stuff like a flour. Like a pizza dough or something like that. You don't have to throw a ton of this stuff at it. You can actually, dilute it and use it at the recommended rates as well, which will make it go a lot further. I'd actually recommend that over using a ton and then hoping it carries over from season to season, because of the benefits of using it on a new seedling for stress and transplant and all the things that we've talked about. But yeah, in terms of carryover, I'd like to see some studies, but I haven't seen any.
Well, I guess I'm wondering is if for example, we had, let's just take a 4x8 raised bed, just to simplify things. And we had a cannabis plant growing in there. And that plant, we go to harvest that plant, and let's say we take out the roots, or the main roots, but don't worry. So there's going to be a bunch of root hairs and leftover root material in that soil. If we were to plant, say, a cover crop or our next cycle of cannabis plants, would the mycorrhizal fungus that had infected the crop before potentially infect the new crop?
Yeah, it's definitely potentially, but it all depends on what species colonize the cannabis and what species of cover crop you're planting and whether or not the two are compatible. It depends on the length of time between the two. If it's a day or two, yeah, it's definitely reasonable and plausible, and I totally support that. I just don't know.
Okay. No, that's fine. I was just curious if you knew the answer.
Yeah, no, I love it. I come from the world of, permaculture and sustainability and environmental science. So I'm all about the least amount of inputs and, extraction and all the, phosphorus mining and fish emulsion that we have to drive 1,000 miles and put on our land. Like, I'm all about growing bioaccumulators and extracting what we can from the land in a sustainable way and reusing it. So yeah, let's do those studies.
So you had mentioned in our email, you have something about explaining techniques for extending effects. Can you touch on that a little bit, or what you meant by that?
Yeah, so like I was talking about with the bacteria, they kind of peak and then plummet. And same thing with the mycorrhizal fungi is the reason that we include sugars in our blends is that those are the preferred, that's the food source of the bacteria and the fungi. That fuels the whole soil food web. So what we've come out with is this product called Mycochum, which has fish hydrolysate, which, as opposed to emulsion, you had the gentleman from Dram on, and I just loved his explanation of how that works. But essentially, the hydrolysate is much better for bacteria and fungi because it contains all of the proteins and fatty acids and a much more diverse palette of foods for these soil organisms. As well, we have kelp, which has a huge array of micronutrients. We have molasses, once again, high in carbohydrates and micronutrients. And then your humic acids, which have your humic substances. So it's feeding the microbes is one thing that I think is really a beneficial thing to do. It's adding your compost teas regularly. It's, topping off your compost teas after you're done brewing them with a little bit of kind of those things that I just talked about to extend their life in the soil. So we've, over the years, the things that we culture and grow these mycorrhizal fungi with, and we've run the gamut of foods to feed these things and to see their response is this is what this Mycochum product is these are all of the foods that the soil food web thrives on. So yeah, what I meant by that was just feeding these organisms, encouraging these organisms. And it's all the same things generally that plants like, right? So you have good aeration, you have a stable pH, you have good water content. You're not overwatering, you're not underwatering, and you're providing a food for these organisms. Things like biochar that provide a habitat, if you will, for these organisms is super beneficial. But really culturing and nurturing these soil organisms is what I was talking about. So you have a healthy population of bacteria that doesn't plummet and then rely solely on the plant exudates. Is you can kind of keep it running at a peak of solubilizing any nutrients in the soil, making available to the plant. So there's like excess of these enzymes and micronutrients available to the plant instead of sort of letting them die back and then there's a bare minimum. So, it's just the same logic as, like I said, the adding compost teas to your soils. You're just kind of maxing out that soil biology so that when the plant needs it, the soil food web delivers.
Okay, that makes sense. I got a copy. I got a sample of the mycochum. I keep wanting to call it chum food. I don't know why I do that. But the mycochum. So the mycochum does not have mycorrhizal fungus in it, but rather it's this combination of fish, kelp, humic, at a very good price point. I'm excited to try it out. My next round of microscope testing, it's at the top of my list of things to experiment with, to see how it works with compost teas and what its effect is on soil microbes. So and I have an order coming in. If there's any listeners that want to try it out, they can reach out to me via email, but it's not available on our website yet. But we can definitely help get some into your hands. I know I'm sending some to my buddy Nelson down in California to do some experiments. So yeah, I'm excited to hear what's going on with that product. But let's go ahead and jump in and talk about the bacteria now and some of these other microbes in your soil, because you and I talked off air about this. I had some questions that you addressed when we had earlier discussions, because one of the things that I've seen is there's a lot of mixed opinions around trichoderma and mycorrhizal fungus. If you look at some of the research, and there's a variety of research out there. One of them was a study at the University of Florida showing that trichoderma inhibited mycorrhizal infection in plants, along with high phosphorus. That one's fairly well established because, one of the main benefits of mycorrhiza is that phosphorus relationship. So if the plant's already getting plenty of phosphorus, it thinks, oh, I don't need this mycorrhizal relationship, even though the mycorrhiza has a bunch of other benefits that you already mentioned for the plant. So we want to avoid high P early on before the plant has time to get that infection. But with trichoderma, there's a lot of different species of trichodermis, which is, I think, important to differentiate. But then in addition to that, it's been shown to inhibit spore germination and colonization of, mycorrhizal fungi, particularly the Endo species. What are sort of your thoughts on that?
Yeah, so I think that, it definitely has been documented that trichoderma can inhibit or can consume mycorrhizal spores, or it can also sort of inhibit the mycelium growth to a degree. I think there's a lot more testing that needs to be done. So there's also other studies that show there's absolutely no effect. There's a study out of Spain that can't see the university, but I'm actually looking at it right now. So they used trichoderma. I can't see the species; it's somewhere in here. But Glomus massae, Glomus deserticola, and they showed. Yeah, I can't find the trichoderma species, but they showed zero effect on colonization. So this study was done in 2000. But yeah, I think there's more research yet to be done. And what we've seen in the studies that we did and in our the research that we accumulated before we made the products is that there's actually a symbiotic, there's a beneficial effect to using both of them. So, there's certain benefits that the trichoderma provide, and then there's certain benefits that the mycorrhizal fungi provide. And, I think when they're used in combination, you actually get a little bit of both. And I don't think it's a black and white thing. I think, like you said, there's there's so many different soil conditions, there's so many different species, and what we base half of our products don't have trichoderma. So we once again, we have the gamut of products. If you're interested in having one with or without, like one of our products, the Orca, the liquid version of Great White, doesn't have trichoderma. So we have products without it, but that being said, Great White's still my favorite product, which has the trichoderma in it. Once again, the benefits of the trichoderma. Well, one of them is just really, really high enzyme release. So this one's breaking down organic matter at a really fast rate compared to other bacteria. But also, like I said, there's benefits that I can't make any claims about. But if you Google trichoderma, there's some biopesticide research about it. So, some people stagger their inoculations, right? So they'll inoculate with the mycorrhizae and then they'll inoculate with trichoderma a little bit later. But one of the things that I like to point out is that lab conditions are so drastically different from field conditions. So some of these studies, documented, oh, hey, this is possible, right? Like, so the trichoderma can become antagonistic towards the mycorrhizal fungi in a petri dish, not necessarily in a growing environment. And one thing about trichoderma is it's absolutely everywhere. So if you ever try and culture like mushrooms indoors, for example, oysters or shiitakes or anything like that, one of the main things that infects your medium is actually trichoderma. So we're breathing it in right now. It's absolutely everywhere, and it's ubiquitous in soils. So it's already there. And the question is, how do these things interact in the soil environment? And from, as far as we can tell, there's a big benefit to actually having it in your medium. And whether or not you stagger the inoculations, that's up to you. But, we've been doing this for 15 years with the Great White, and that's the product that has it in there. And growers just have tremendous results. I think we've seen a benefit, and in using both in combination. I think in if there is a situation where it's becoming antagonistic to the mycorrhizal fungi, I don't know. I feel like people are sometimes splitting hairs over this. Like, clearly using a product like Great White with trichoderma outperforms non-inoculation every time. And even when compared with, say, for example, a single species mycorrhizal versus a multiple species mycorrhizae with a multiple species bacteria and trichoderma, it just performs fantastic. So I think there's a lot more research yet to be done, and I don't think it's black and white. I think Elaine Ingham is a really amazing researcher, and a lot of her actual field knowledge of how this stuff performs in actual soil conditions is very valuable. And from the testing that we've done and the feedback that we've gotten from our friends and growers is that it works really well. So, I don't think the jury's in on that. But like I said, I have access to all of the products here at the company, and I consistently, Great White for me is the pinnacle. So.
Yeah, you brought up a few different points I want to touch on here. So it's pretty well established that trichoderma attacks mycorrhiza in plate assays, like you mentioned. But in soils, like I've seen some studies that show it does cause damage to the mycorrhizal infection rates, but there's also other ones that show that maybe the spatial distribution in soils allow both to function normally or to have a synergistic positive interactions. So I think you really need to experiment with it. And then ultimately, you might be right that, yes, if it is the trichoderma that is providing the benefit, like that's something that Tim Wilson, the microbe-organics, has speculated for years. At the end of the day, does it really matter if you're getting a plant growth response or plant health response that you want? Anyway, does it matter if it was the trichoderma or the mycorrhizal infection that's providing these benefits? One thing I've always wondered about, though, is really, so like you mentioned, trichoderma is ubiquitous. It's an aggressive microbe. When you're putting in, say, 187,000 CFUs per gram, how does a Endomycorrhiza at, a mere 200 spores per gram or propagules per gram really compete in that space. That's one question I've always had about is sort of the difference in concentrations. I know trichoderma is a lot more affordable to add into these products, but why the different discrepancy in the rates, I guess?
Yeah, so one thing is that the great white in testing has shown great mycorrhizal colonization of the root system. So it's an effective product, and the mycorrhizae is still infecting the plants at a successful rate. In terms of the question, I'd have to ask the researchers on that, or our tech guys, because I don't know the answer of that. And like you said, that research of the spatial distribution is important to allow each to sort of function and not cannibalize, eat the mycorrhizae. So yeah, that's a good question about the concentrations. But yeah, like I said, at this point, the product is successful and works and has a high colonization rate, but I don't know how that actually plays out with the ratio there. So that's a good question. Another one I'll have to do more research on.
Okay, and to touch on some of these other bacteria that you have in the product that I recognize, like Bacillus subtilis, that's in Serenade and a bunch of other products. Again, particular species of Bacillus subtilis, but that's another one that's been shown to have a lot of different plant benefits. The Bacillus thuringiensis, I can never pronounce it, the BT, the Israelis species is one that's super useful when trying to control fungus gnats and other issues like that, or caterpillars. It's something you'd spray on. So that has a lot of benefits. And then I noticed you have Saccharomyces cerevisiae, which is brewer's yeast, essentially in there. Is that I know people are taking barley seeds and grinding those up as a way of as something also from the brewing industry, and I've read some really wonderful books on that particular fungus. But do you happen to know much more about what benefits that has in the soil?
So that species specifically, I'm not a bacteria expert. I will say that at the get-go. I don't have actually a ton of research on the Saccharomyces cerevisiae besides the beer that I brew. You probably actually are more of an expert on that than I am. So, but bacteria in general, you kind of already cover it. But, the sort of, a lot of them do a lot of the same things, just kind of from a different species, different angle. But in general, you're getting, nitrogen fixation, phosphorus solubilization, micronutrient solubilization, and enzyme production. They also secrete organic glues, like high carbon glues that help with soil structure over the long term. And so, those are the big benefits, and that really works in synthesis or in combination with the mycorrhizal fungi and help with their needs and their sort of ability to extract these micronutrients from the soil. So, we include them. We're more specialized. We cut our teeth and we started in the mycorrhizal space and, slowly included bacteria in our products. So like I said, I'm not an expert on the bacteria, but those are just sort of an overview of what they do and what they add to our microbe products.
Okay. I know there's a lot we don't know as a species about all of these things. Like humans are still learning so much about all of these different microbes and how they interact and discovering new microbes. And so I don't expect you to have all the answers by any means.
Right.
But we are seeing, with all of this research, that diversity really is one of the most important things when it comes to soil health. I can see why you would want to be applying a diversity of microbes when you're inoculating your soil.
Yeah. Yeah, one thing that blows my mind is like the microscope has only been around for 100 years and popular use for less than that. So, this whole world of microbes is just coming into focus for us as a species. And, there's hundreds of thousands of species out there that aren't tested. Your average compost bin is like, talk about diverse. These are just the species that we've isolated and actually ran through the gamut of expensive testing to see their benefits on agriculture and plant growth. But this is just the tip of the iceberg. There's so much extra. There's so much diversity out there that we don't know about. And in general, like you said, it's about diversity, and it's about sort of doing those things like composting and compost teas and all those practices that, in my mind, they mimic natural systems, right? This is how the forest is one of the most productive ecosystems on the planet, and it's all passive, right? Everything leads into something else. Nothing's wasted. So a lot of what we're doing, in my mind, we're learning more about how these systems function, but a lot of what I do and what we do as a company is really just trying to mimic the best of these natural systems and their efficiency. So, yeah, there's a lot left to learn.
I totally agree, and I love the idea of trying to just balance or recreate these ecological systems, like you said, that we know are highly functional and sustainable. So, yeah, thanks for coming on the show today. Was there anything else that I'm out of questions? Was there anything else you wanted to touch on, though, before we go?
No, I think that pretty much covers it.
All right. Well, hey, I really appreciate your time today. Thanks again. And I look forward to meeting you in person and talking to you again soon.
Thanks, Tad. Yeah, I really enjoyed it. You can follow us on all the traditional social media channels at Great White Myco or Plant Success Organics. So if people are interested in getting more information, reach out, send us a message, and we'd be happy to help you out, answer any questions, and spread the good word.
That was Jim Eagan with Plant Revolution. I posted the links and information we discussed in this podcast right on the podcast page at www.kisorganics.com. Just click on the podcast menu on the top of the home screen. And don't forget to sign up for our newsletter on our website right on the homepage and subscribe on your favorite listening platform so you can stay up to date with all the latest information and podcasts right when they come out. You can also view and download the new white papers and their results on our website blog page. I've gotten some great feedback on them since posting and hope to get Jaya Palmer back on the podcast soon to discuss his methodology and how he set up the trial. Thanks for listening.